summer workshop. My name is Judy, and another host today are Shivika, Nivi, Ashley, Cozy, and Michelle. During today's workshop, we're going to discuss a lot about COVID-19, clinical research, medical school admissions, and minorities representation in the medical field. Today's agenda is going to be like this. From 10:30 to 10:40 Eastern Daylight Time is the opening ceremony, which is currently right now. And from 10:40 to 11:30, members from Revolutionize STEM will broadly cover the topics of COVID-19 basics, including its symptoms, the virus variants, COVID-19 test, what is a vaccine, different types of COVID-19 vaccines, and how do they protect us from the virus, myths around virus and vaccines, and research about susceptibility to COVID-19 virus. Then, from 11:30 to 12:15, we're joined by a guest speaker who is a research fellow in biological chemistry and molecular pharmacology at Harvard Medical School, and she'll be discussing clinical research, epidemiology, and COVID-19 as a case study. From 10:15 to 10:45, we're going to have a 30 minutes break. Then coming back from 10:45 to 1:45, we're joined by another guest speaker who graduated from the University of Pennsylvania and MIT and is currently a professor and chair of science education at Zucker School of Medicine. She'll be discussing medical school admissions, importance of minorities in the medical field, and the evolution of medicine throughout the years. Then from 10:50 to 20, we will welcome two youth-led nonprofit organizations who is each going to make a 15 minutes presentation about their mission, goal, and achievements. At the end, from 2:20 to 2:30, we will have the end note and wrap up our summer workshop. Certificates will be given out to all participants, and we will also send out a feedback form to collect suggestions for further improvement. That's the overview of our workshop, and we're thankful again for you all to join us. Now I'm going to turn the speaker to the Revolutionize STEM members who will discuss the COVID-19 basics.
Hi everyone, we'll be going over the COVID-19 basics, the symptoms of COVID-19 and the various variants, the COVID test and the vaccines, and also the different vaccines and how they protect us from COVID, myths about the COVID-19 virus, myths about the vaccine, and the research on susceptibility to COVID-19.
So to start, here's a quote from Marie Curie, who is a Polish-French physicist who looked into radioactivity. "Nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less." So here is a timeline of the coronavirus pandemic. So it was on January 9th when the WHO announced findings of a mysterious coronavirus-related pneumonia, and this was in Wuhan, China. It then went on January 21st, where a scientist confirmed that it could be transferred from person to person. With a worldwide death toll of more than 200 and an exponential jump to more than 9,800 cases, the WHO finally declares a public health emergency for just the sixth time. In declaring the COVID-19 a pandemic, Tedros Adhanom Ghebreyesus, Director General of the WHO, said at a briefing in Geneva, the agency is deeply concerned by the alarming levels of spread and severity of the outbreak. In an updated scientific brief, WHO notes that the virus may linger in the air in crowded indoor spaces and emphasizes that the virus may be spread by asymptomatic individuals. So COVID-19 comes from a family of viruses known as coronavirus, which cause a lot of colds and upper respiratory infections. SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2. People infected can be asymptomatic, show no symptoms while infected, but can still transmit the virus, the same as any person showing symptoms, or even show a range of symptoms from severe to mild. The average time for the incubation period, the time from when someone is infected when they show symptoms for COVID, is 3 to 5 days, but symptoms can actually show up 13 days after infection. COVID-19 is spread person to person through aerosols, a person would emit, which could be inhaled by other people, which would infect them. Aerosols are infectious viral particles that can float or drift around in the air for up to 3 hours. Droplets that are produced when an infectious person coughs or sneezes may land in the mouths or noses of people who are nearby and possibly inhaled into the lungs. This can also happen while they're talking. So some common symptoms are fever, body aches, dry cough, fatigue, chills, headache and sore throat, loss of appetite. There are also more rare symptoms that can be a negative impact on brain function, which can mean loss of smell, inability to taste, muscle weakness, tingling or numbness in the hands and feet, dizziness, confusion, delirium, seizures, and stroke. And some people also have gastrointestinal issues, such as loss of appetite, nausea, vomiting, diarrhea, and abdominal pain or discomfort associated with COVID-19. So here is a breakdown of some of the different variants of COVID-19. We start with the Alpha variant, which is from the UK. There is a 50% increase in transmission, potential increased severity, and no impact on the success of antibody treatment. There is then the Beta variant, which is from South Africa, a 50% increase in transmission, and it significantly reduces susceptibility to some antibody treatments. There's the Gamma variant, originally in Japan and Brazil, and this significantly reduces susceptibility to some antibody treatments. And then we have the Delta variant, which originated in India, which caused a large increase in transmission. There is less research due to the novelty, but it can be really deadly, and it's more common towards the younger ages. So here's a slide about COVID tests. Now, there are two different types of COVID-19 tests. There's a diagnostic test. Molecular and antigen types of diagnostic tests are used to test people for COVID-19. Samples are usually collected through nasal or throat swabs or saliva and collected by spitting in a tube. Molecular tests take less than 1 hour or 1 to 3 days. Antigen tests take 15 to 30 hours. Antibody tests. This test looks for antibodies in your immune system which would have been produced to fight off COVID-19. They cannot be used to diagnose current infections, but can be used to discover past ones. Samples come from blood drawn on a finger stick or blood drawn by a doctor. This test is useful for asymptomatic people who may have had COVID in the past.
So what are vaccines? Vaccines are a substance used to provide immunity from one or several diseases. The importance of vaccination. It is effective at preventing the spread of COVID-19 and can keep individuals from getting COVID. It also prevents vaccinated people from getting seriously ill with COVID, and the herd immunity would protect whole communities. People's opinions about vaccination. Anti-vaccine people who have anti-vaccination opinions believe myths and conspiracy theories about the vaccine and refuse to get it, as well as some religious reasons. Pro-vaccine. They trust the science behind the vaccine and most likely would get it. There are different types of vaccines that companies have used for the COVID-19 vaccine. The first is the mRNA. This type of vaccine uses genetically engineered mRNA to give your cells instructions for how to make the S protein found on the surface of the COVID-19 virus. After vaccination, your immune cells begin making the S protein pieces and displaying them on cell surfaces. This causes your body to create antibodies. If you later become infected with the COVID-19 virus, these antibodies will fight the virus. After delivering instructions, the mRNA is immediately broken down. It never enters the nucleus of your cells where your DNA is kept. Both the Pfizer biotech and the Moderna COVID-19 vaccine use mRNA. Protein-based vaccines. These vaccines include only parts of a virus that best stimulate your immune system. This type of COVID-19 vaccine contains harmless S proteins. Once your immune system recognizes the S proteins, it creates antibodies and defensive white blood cells. If you later become infected with the COVID-19 virus, the antibodies will fight the virus. The third type is the viral vector. In this type of vaccine, genetic material from the COVID-19 virus is placed in a modified version of a different virus, hence viral vector. When the viral vector gets into your cells, it delivers genetic material from the COVID-19 virus that gives your cells instructions to make copies of the S protein. Once your cells display the S proteins, once your cells display the S proteins on their surfaces, your immune system responds by creating antibodies and defensive white blood cells. If you later become infected with the COVID-19 virus, the antibodies will fight the virus. Viral vector vaccines can cause you to become infected with the COVID-19 virus or the viral vector virus. Also, the genetic material that's delivered doesn't become part of your DNA. Let's look into some of the vaccines developed. First one is the Pfizer vaccine. The, the Pfizer vaccine is an mRNA vaccine that has to be stored at negative 80 degrees Celsius to negative 60 degrees Celsius for 6 months or 2 to 8 degrees Celsius for up to 5 days, and it needs to be given in 2 doses. For the Moderna vaccine, it is also an mRNA vaccine that has to be stored at negative 25 degrees Celsius to negative 15 degrees Celsius for 6 months and 2 to 8 degrees Celsius for up to 30 days. It also has to be given in 2 doses. For the Janssen vaccine, it is a viral vector vaccine that has to be stored in 2 to 8 degrees Celsius for 3 months and only needs 1 dose. The AstraZeneca vaccine is also a viral vector vaccine. It needs to be stored at 2 to 8 degrees Celsius for up to 6 months, and it has to be given in 2 doses. The Novavax vaccine. It is a protein-based vaccine, and it has to be stored at 2 to 8 degrees Celsius and has no time limit on how long it can be stored. And it also has to be given in 2 doses. The Sputnik 5 is a viral vector vaccine. It has to be stored in negative 18.5 degrees Celsius in liquid form, but 2 to 8 degrees Celsius in dry form. This vaccine also has to be given in 2 doses. The most used vaccines are Pfizer, which is used in 111 countries, Moderna, which is used in 61 countries, AstraZeneca, which is used in 118 countries. The effectiveness of each vaccine. Pfizer is effective for 97%. Pfizer has an efficacy of 97%, Moderna 95%, Janssen & Janssen 70, AstraZeneca 74, Novavax has one of 91, and Sputnik 5 is again around 95%. So for some secondary effects of the vaccines, AstraZeneca can have headache, joint pain, and muscle pain. Moderna can have headache, joint pain, swelling, and muscle pain. And Pfizer could be headache, diarrhea, or muscle pain. AstraZeneca could have tiredness, shaking, chills, or fever. Moderna could have sickness, vomiting, acute tiredness, shaking, chills, and fever. Pfizer could have fatigue, malaise, shaking, chills, feeling of fever, and sickness. Give me 1 second, the slides are stuck. Okay. So the vaccine phases that most countries are using are phase 1, where the healthcare workers get the vaccine, phase 2, where essential workers and high-risk population get the vaccine, and then phase 3 goes from oldest to youngest people who are eligible who can get the vaccine. There's a possibility for a vaccination passport to travel internationally and domestically to contain the spread of COVID-19. 3.79 billion vaccine doses have been administered around the global population.
So now we're going to be talking about common myths about the COVID virus. So fact, being able to hold your breath for 10 seconds or more without coughing or feeling discomfort does not mean you are free from the COVID-19 virus. Symptoms of the COVID-19 include dry cough, tiredness, and fever. Sometimes people may develop more severe forms of the disease, such as pneumonia. The best way to confirm if you have COVID-19 is with a laboratory test. You cannot confirm it with a breathing exercise, which can even be dangerous. Fact, most people who get the COVID-19 recover from it. Most people who get it have mild or moderate symptoms and can recover thanks to supportive care. If you have a cough, fever, or difficulty breathing, seek medical care early. Call your health facility by telephone first. If you have a fever and live in an area with malaria or dengue, seek medical care immediately. If you are a high-risk person, then seek medical care urgently. Fact, ordering or buying products shipped from overseas will not make a person sick. The likelihood of becoming infected with COVID-19 from a commercial package is low, since it has likely traveled over several days and been exposed to different temperatures and conditions during transit. Fact, people should not wear masks while exercising. People should not wear masks when exercising, as masks can reduce the ability to breathe comfortably. Sweat can make the mask more wet, more quickly makes it difficult to breathe, and promotes the growth of microorganisms. The important preventive measure during exercise is to maintain physical distance of at least 1 meter from others. Fact, the COVID-19 was not deliberately created or released by people. Viruses can change over time. Occasionally, a disease outbreak happens when a virus that is common in animals, such as a pig, bat, or bird, undergoes changes and passes to human. This is likely how the new coronavirus came to be. Fact, no one or ethnic, racial, gender, or religious group of people is more likely to get or transmit the virus. There have been hate crimes committed all over the world due to the fear that the Chinese brought the virus and are most likely to transmit it. This is not true. Many Asians lost their lives, and even more have been injured. Okay, so now we'll go over the most common myths about the COVID vaccine. Okay, both fact, a COVID-19 vaccine will not alter your DNA. COVID-19 will not change or interact with your DNA in any way. Both mRNA and the viral vector COVID-19 vaccines deliver instructions or genetic material to our cells to start building protein against the virus that causes the COVID-19. However, the material never enters the nucleus of the cell, whereas your DNA is kept. Fact, COVID-19 vaccine does not cause people to test positive for COVID-19 on the viral test. A viral test are used to see if you have a concurrent infection. If your body develops an immune response to the vaccination, which is the goal, you may test positive on some antibody tests. Antibody tests indicate you have a previous infection and that you may have some level of protection against a virus. Fact, if you have already had COVID-19, you should still get the vaccine. Due to the severe health risks associated with COVID-19, the fact that reinfection with COVID-19 is still possible, there is not enough information currently available to say if or for how long people are protected from getting the COVID-19 after they've had it, which is called natural immunity. Early evidence suggests natural immunity from COVID-19 may not last very long, but more studies are needed to better understand this. Several subjects in the Pfizer trial who were previously infected got vaccinated without ill effects. Sometimes some scientists believe the vaccine offers better protection for coronavirus than natural infection. Fact, although researchers rushed to develop the COVID-19 vaccine, its effectiveness and safety can still be trusted. Studies found that the two initial vaccines, Moderna and Pfizer, are both about 95% effective and reported no serious or life-threatening side effects. There are many reasons why the COVID-19 was developed so quickly. Let's go over some. The COVID-19 vaccines, both Pfizer, BioNTech, and Moderna, were created with a method that has been developed for years, so company could start the vaccine development process early in the pandemic. China isolated and shared genetic information about the COVID-19 promptly so scientists could start working on the vaccines. The vaccine developers didn't skip any testing steps, but conducted some of the steps on an overlapping schedule to gather data faster. Vaccine projects had plenty of resources as governments invested in research and/or paid for vaccines in advance. Some types of COVID vaccines were created using messenger RNA or mRNA, which allows faster approach than the traditional way that the vaccines are made. Social media helped companies find and engage study volunteers, and many were willing to help with COVID-19 vaccine research. Because COVID-19 is so contagious and widespread, it did not take long to see if the vaccine worked for study volunteers who were vaccinated. Companies began making vaccines early in the process, even before FDA authorization, so some supplies were ready when authorization occurred. Fact, after getting the COVID-19 vaccine, you can stop wearing a mask. On May 13, 2021, the CDC revised safety guidelines for those who are fully vaccinated against the coronavirus, stating that fully vaccinated people can resume activities without wearing a mask or physically distancing. The CDC continues to recommend that masks and physical distancing are required when going to doctor's office, hospitals, or long-term care facilities, but you should still be taking COVID precautions as they are in your best interest. Okay, fact. Getting the COVID vaccine does not give you COVID. The vaccine for COVID-19 cannot and will not give you COVID-19. The two authorized mRNA vaccines instruct your cells to produce a protein that is part of the SARS-CoV-2 coronavirus, which help your body recognize and fight the virus if it comes along. The COVID-19 vaccine does not contain the SARS-CoV-2 virus, so you cannot get COVID-19 from the virus. The protein that helps your immune system recognizes and fights the virus and does not cause infection of any sort. Fact, the side effects of COVID-19 vaccine are not severely dangerous. The Pfizer and Moderna COVID-19 vaccines can have short side effects, but the vast majority are very short-term, not serious or dangerous. The vaccine developers report that some people experience pain when they're injected, body aches, headaches, or fever, lasting a day or two. These are signs that the vaccine is working to stimulate your immune system. If symptoms persist beyond 2 days, you should call your doctor. If you have allergies, especially severe ones that require you to carry an EpiPen, discuss the COVID-19 vaccine with your doctor, who can access your risk and provide more information about if and how you can get vaccinated safely.
Fact: just because we now have a vaccine for COVID-19 does not mean we can make vaccines for the common cold, HIV, and other diseases. The thousands of viruses that cause various diseases are very different. Many change or mutate year by year, making it difficult to develop one vaccine that works for a long period of time. Developing vaccines for some disease-causing viruses is tough. For example, the virus that causes HIV can hide and make itself undetectable by the human immune system, which makes creating a vaccine for it extremely difficult. The common cold can be caused by any one of hundreds of different viruses, so a vaccine for just one of them would not be very effective. Research on transmission of COVID-19. Look at transmissibility, specifically among asymptomatic and pre-symptomatic patients. We first have to define asymptomatic and pre-symptomatic and distinguish the meaning of both. Asymptomatic. An asymptomatic individual is defined as someone who tests positive for the virus but lacks symptoms that would indicate an infection. An asymptomatic person may go through the entire course of virus infection and never experience any symptoms. Pre-symptomatic. An individual that initially presents as asymptomatic but goes on to develop symptoms days or weeks later after the initial infection. And here's a quote: "We know very little about the proportions of people with positive results who are truly asymptomatic throughout the course of their infection." Finally, the transmission of COVID-19. Testing for the virus can detect whether or not a virus is present in an individual, but it doesn't tell us if that person is contagious. A crucial mechanism in the transmissibility of COVID-19 is the high level of the SARS shedding in the upper respiratory tract.
Both groups can spread the infection. The role of symptomatic, pre-symptomatic, and asymptomatic transmission have in the spread of SARS-CoV-19 is still debated on. Some scientists argue that asymptomatic persons play a major role in the transmission of COVID-19. Others argue that it has a lesser role than symptomatic and pre-symptomatic transmission. Now we have a video that we're going to be showing.
This is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles, which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gather just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane. Now, with the virus spreading in your body, how can you develop pneumonia symptoms? For this, we'll have to look into your lungs. Each lung has separate sections called lobes. Normally, as you breathe, air moves freely through your trachea or windpipe, then through large tubes called bronchi, through smaller tubes called bronchioles, and finally into tiny sacs called alveoli. Your airways and alveoli are flexible and springy. When you breathe in, each air sac inflates like a small balloon. And when you exhale, the sacs deflate. Small blood vessels called capillaries surround your alveoli. Oxygen from the air you breathe passes into your capillaries, and then carbon dioxide from your body passes out of your capillaries into your alveoli so that your lungs can get rid of it when you exhale. Your airways catch most germs in the mucus that lines your trachea, bronchi, and bronchioles. In a healthy body, hair-like cilia lining the tubes constantly push the mucus and germs out of your airways, where you might expel them by coughing. Normally, cells of your immune system attack viruses and germs that make it past your mucus and cilia and enter your alveoli. However, if your immune system is weakened, like in the case of a coronavirus infection, the virus can overwhelm your immune cells, and your bronchioles and alveoli become inflamed as your immune system attacks the multiplying viruses. The inflammation can cause your alveoli to fill with fluid, making it difficult for your body to get the oxygen it needs. You could develop lobar pneumonia, where one lobe of your lungs is affected, or you could have bronchopneumonia that affects many areas of both lungs. Pneumonia may cause difficulty breathing, chest pain, coughing, fever and chills, confusion, headache, muscle pain, and fatigue. It can also lead to more serious complications. Respiratory failure occurs when your breathing becomes so difficult that you need a machine called a ventilator to help you breathe. These are the machines that save lives and that medical device companies currently ramp up production for. Whether you would develop these symptoms depends on a lot of factors, such as your age and whether you already have an existing condition. While this all sounds scary, the push to develop a coronavirus vaccine is moving at high speed. Studies of other coronaviruses led most researchers to assume that people who have recovered from a SARS-CoV-2 infection could be protected from reinfection for a period of time. But that assumption needs to be backed by empirical evidence, and some studies suggest otherwise. There are several different approaches for a potential vaccine against the coronavirus. The basic idea is that you would get a shot that contains faint versions of the virus. The vaccine would expose your body to a version of the virus that is too weak to cause infection, but just strong enough to stimulate an immune response. Within a few weeks, cells in your immune system would make markers called antibodies, which would be specific for only the coronavirus, or specifically its spike protein. Antibodies then attach to the virus and prevent it from attaching to your cells. Your immune system then responds to signals from the antibodies by consuming and destroying the clumps of viruses. If you then catch the real virus at a later stage, your body would recognize it and destroy it. In other words, your immune system is now primed. Collecting evidence on whether this will be possible, safe, and effective is part of what's taking researchers so long to develop a vaccine. It's a race against time to develop a vaccine amid a pandemic. Each step in vaccine development usually takes months, if not years. An Ebola vaccine broke records by being ready in 5 years. The hope here is to develop one for the new coronavirus in a record-breaking 12 to 18 months. While all this will take time, stay home if you can to protect the most vulnerable. And don't forget to wash your hands for at least 20 seconds and as often as possible.
That was a video about the COVID-19 virus and kind of an animation of how this virus works and infects people.